Deep within the nucleus of every human cell lies a microscopic marvel, a molecule so intricate it holds the complete blueprint for building and operating your entire body. This is deoxyribonucleic acid, or DNA, a double helix that stretches nearly 2 meters when unraveled, yet packs into a space just 6 microns wide. Each strand is composed of 4 chemical bases, adenine, thymine, cytosine, and guanine, arranged in sequences that act like a biological language. The order of these bases determines everything from your eye color to your susceptibility to certain diseases. Remarkably, if you were to type out the entire human genome at 60 words per minute, it would take you about 50 years, non-stop. DNA is not merely a static molecule, it is a dynamic, self-replicating archive that has been refined over billions of years of evolution. The discovery of DNA's structure is one of the most celebrated stories in science. In 1953, James Watson and Francis Crick, using X-ray diffraction images from Rosalind Franklin, unveiled the double helix model. Franklin's photo 51 revealed a clear X-shaped pattern, proving the molecule had a helical structure. Watson and Crick's model showed 2 antiparallel strands held together by hydrogen bonds between complementary base pairs. This elegant architecture immediately suggested how DNA could replicate, the strands unzip, and each serves as a template for a new partner. The Nobel Prize was awarded to Watson, Crick, and Wilkins in 1962, but Franklin's pivotal contribution was recognized only posthumously. Today, the double helix stands as an icon of molecular biology, representing the very code of life. Every time a cell divides, its DNA must be copied with astonishing fidelity, a process called replication. An enzyme named DNA polymerase zips along the strand, reading the template and adding matching bases at a rate of about 1,000 per second. Yet mistakes happen roughly once every 10 billion bases, a rate so low that only about 1 to 3 errors occur per cell division. Repair enzymes immediately correct these typos, preventing catastrophic mutations from accumulating. However, if a mistake slips through, it can become a permanent change, a mutation, which may be harmless, beneficial, or harmful. This precise copying mechanism is why we inherit so much from our parents, yet also why evolution can occur over generations. Replication is the engine of heredity, ensuring life's continuity while allowing room for change. Genes are the functional units of DNA, specific segments that contain instructions for making proteins. Humans have about 20,000 protein-coding genes, which is surprisingly similar to the number in a simple worm. But our complexity comes from how these genes are regulated and spliced. A single gene can produce multiple protein variants through alternative splicing, vastly expanding the proteome. Each gene is preceded by a promoter region that acts like a switch, controlling when and where the gene is activated. Mutations in a gene can disrupt protein function, leading to disorders like cystic fibrosis or sickle cell anemia. Yet many variations are completely harmless, contributing to the beautiful diversity of human traits. Understanding genes is like reading the operating manual of our biology. Mutations, changes in the DNA sequence, are the raw material of evolution and the cause of many genetic diseases. They can arise from errors during replication, exposure to radiation, or chemicals that damage DNA. A single base substitution can have profound effects. The sickle cell mutation changes just 1 amino acid in the hemoglobin protein, yet it alters red blood cells from disc-shaped to crescent-shaped. This causes severe pain and organ damage, but also provides resistance to malaria. Not all mutations are bad, some are neutral, and rare beneficial ones give an evolutionary advantage. Over generations, natural selection filters these variations, driving adaptation. Our DNA carries the scars and gifts of millions of years of mutational history. Each of us is a unique mosaic of inherited and new mutations. The genetic code is the universal language that translates DNA into proteins. Every 3 bases, a codon, specifies one of 20 amino acids. With redundancy built in, most amino acids are encoded by several codons. This code is nearly identical in all life forms, from bacteria to humans, evidence of a common ancestry. Ribosomes, the molecular machines inside cells, read messenger RNA, a copy of a gene, and assemble amino acids into a chain to form a protein. The code was deciphered in the 1960s by Marshall Nirenberg and others, who used synthetic RNA sequences to crack the triplet system. The discovery revealed that life's instructions are written in a simple, elegant language. This code is the Rosetta Stone of biology, unlocking the secrets of how DNA builds the proteins that run our bodies. Epigenetics reveals that DNA is not destiny. Environment and lifestyle can influence gene activity without changing the underlying sequence. Chemical marks, like methyl groups attached to DNA, can switch genes on or off. These epigenetic modifications can be inherited, passing from parent to child, and can be altered by diet, stress, and exposure to toxins. For example, identical twins share the same DNA, but epigenetic changes accumulated over their lives can make them increasingly different in health and appearance. Understanding epigenetics offers new hope for treating diseases like cancer, where abnormal methylation silences tumor suppressor genes. It also explains how the same genome can produce hundreds of different cell types, from neurons to skin cells, by selectively activating only relevant genes. Epigenetics adds a layer of complexity that challenges the simple view of DNA as a rigid blueprint. The Human Genome Project, completed in 2003, was a monumental international effort to sequence all 3 billion base pairs of human DNA. Led by scientists at 20 institutions across 6 countries, it took 13 years and cost about $2.7 billion. The resulting reference genome is a digital map of our genetic heritage, revealing the location and sequence of every gene. This project has revolutionized medicine, enabling researchers to pinpoint genes linked to diseases like breast cancer, BRCA1 and BRCA2, and to develop targeted therapies. It also unveiled that humans share about 98.8% of their DNA with chimpanzees and even 60% with fruit flies. The Human Genome Project didn't just sequence DNA, it opened the door to the era of personalized medicine. Today, you can get your own genome sequenced for under $1,000. One of the biggest surprises from the Human Genome Project is that protein-coding genes account for only about 2% of our DNA. The remainder, once dismissed as junk DNA, is now known to be vital for regulation, structure, and evolution. This non-coding region includes elements that control gene expression, such as enhancers and silencers, as well as repetitive sequences that help maintain chromosome integrity. Some non-coding DNA produces functional RNA molecules that regulate other genes. Recent research shows that many diseases, including cancer, involve mutations in these non-coding regions. The term junk DNA has been mostly abandoned. Instead, scientists speak of a dark genome full of undiscovered functions. This vast unknown reminds us how much there is still to learn about our own genetic makeup. The future of DNA science is nothing short of astonishing. CRISPR-Cas9 gene editing allows us to precisely cut and modify DNA, opening the door to correcting genetic defects. In clinical trials, CRISPR is being used to treat sickle cell disease and certain types of blindness. Long-read sequencing technologies are revealing previously hidden structural variations in our genomes. We are also beginning to understand how DNA functions in three-dimensional space inside the nucleus, with loops and folds bringing distant genes into contact. Synthetic biology aims to design entirely new genomes, potentially creating organisms with novel capabilities. The secrets of human DNA are being unlocked rapidly, promising a future where genetic diseases may become curable, and our understanding of life itself will deepen immeasurably. The double helix has only just begun to share its secrets. But what makes you uniquely you? The answer lies in a tiny fraction of your DNA, less than 1% of your entire genetic code. This minuscule variation, scattered across 3 billion base pairs, holds the secrets to your eye color, your height, your susceptibility to certain diseases, and even your ability to digest milk. Scientists call these differences single nucleotide polymorphisms, or SNPs for short. Each SNP represents a single letter change in the DNA alphabet, a tiny mutation passed down through generations. These variations are the brushstrokes that paint the portrait of human diversity, making each of us a one-of-a-kind masterpiece. To understand how these tiny changes shape our lives, we must travel back in time. Imagine a world where our ancestors first migrated out of Africa, braving new climates, foods, and diseases. Those who survived passed on their genetic adaptations, like the ability to digest lactose in adulthood, a trait that emerged just 10,000 years ago with the rise of dairy farming. Today, this SNP is common in Northern Europeans but rare in East Asians. Your DNA is a living fossil, a record of your ancestors' struggles and triumphs, etched into every cell of your body. But DNA variation isn't just about history, it's a matter of life and death. Consider the BRCA1 gene, where certain SNPs dramatically increase the risk of breast and ovarian cancer. A single letter change can turn a tumor suppressor into a ticking time bomb. Yet, the same gene can also be a lifesaver. In Iceland, a specific BRCA2 mutation actually protects against some cancers. This duality reveals the delicate balance of our genetic code, where one mutation can be both a curse and a blessing, depending on the environment and other genes. Now, let's zoom in on a single SNP that changed the world, the mutation for sickle cell disease. This tiny change in the hemoglobin gene causes red blood cells to deform into crescent shapes, leading to pain and organ damage. But there's a dark twist. In regions plagued by malaria, carriers of this mutation have a survival advantage. The sickle-shaped cells make it harder for the malaria parasite to infect them. This is evolution in action, a brutal trade-off where a deadly disease protects against an even deadlier one, a genetic bargain struck by natural selection. But not all genetic variation is harmful. Some SNPs give us superpowers. Take the ACTN3 gene, nicknamed the speed gene. A specific variant is linked to explosive muscle power, found in elite sprinters and power athletes. In fact, 95% of Jamaican sprinters carry this variant, compared to 50% of Europeans. Meanwhile, another SNP in the EPAS1 gene allows Sherpas to thrive at high altitudes with less oxygen. Your DNA might hold the key to your hidden talents, a genetic lottery that influences everything from your athletic potential to your tolerance for spicy food. But how do we find these life-altering SNPs? The answer is genome-wide association studies, or GWAS. Scientists scan the DNA of thousands of people, comparing those with a trait to those without. They look for SNPs that appear more often in one group. It's like finding a needle in a haystack, but with supercomputers and big data. GWAS has linked SNPs to everything from height and hair color to schizophrenia and heart disease. Yet, each SNP usually has a tiny effect, and it's the combination of thousands that shapes who we are. The most famous GWAS discovery is the FTO gene, the fat gene. A specific SNP in FTO increases the risk of obesity by 30%. But here's the twist, the effect is not about metabolism but behavior. This SNP makes people crave high-calorie foods and feel less full after eating. It's a genetic predisposition, not a destiny. Knowing this, scientists are developing personalized diets that bypass these cravings. Your DNA doesn't just predict your future, it can help you rewrite it, one meal at a time. But the power of SNPs extends beyond health. They are rewriting history. By analyzing ancient DNA, scientists can trace human migrations with stunning precision. A single SNP in the Denisovan genome, found in modern Tibetans, allows them to live at high altitudes. Another SNP from Neanderthals affects our immune system and even our risk of depression. Your DNA is a mosaic of ancient encounters, a living library of extinct species that once walked the earth alongside our ancestors. Each SNP is a whisper from the past. Now, for the first time, we can edit these SNPs. CRISPR-Cas9, a molecular scissors, can cut and replace a single letter of DNA. Scientists are already using it to correct the sickle cell mutation in human patients, with stunning results. But this power comes with ethical dilemmas. Should we edit the genes of embryos? Could we create designer babies with enhanced traits? The technology is racing ahead of the debate, forcing humanity to confront a new frontier. Your DNA is no longer destiny, it's a draft, waiting for revision. As we stand at this genetic crossroads, one thing is clear, your DNA is not a fixed blueprint but a dynamic story. Every SNP is a chapter, written by your ancestors, edited by your environment, and now, potentially rewritten by you. The secrets of human DNA are no longer hidden, they are in our hands. From the ancient migrations that shaped our species to the CRISPR edits that will define our future, the tiny variations in your genome hold the power to heal, to understand, and to transform. The question is, what story will you write? Deep within the nucleus of nearly every cell in your body lies a molecular masterpiece, your DNA. This double helix is not a static blueprint, but a dynamic instruction manual, tightly coiled and exquisitely organized. Each strand is composed of just 4 chemical bases, adenine, thymine, cytosine, and guanine, repeating billions of times. The specific sequence of these bases encodes the information to build and maintain your entire body. If you were to stretch out all the DNA from a single human cell, it would extend about 2 meters in length. Now imagine the entire genome, all 3 billion base pairs, packed into a nucleus just 6 microns wide. This is biological engineering at its most breathtaking and efficient. But how does this microscopic ladder actually read its own instructions? The key lies in a process called transcription, where a specialized enzyme, RNA polymerase, unzips a segment of the DNA. It then uses one strand as a template to build a complementary molecule of messenger RNA. This mRNA acts as a portable copy, leaving the nucleus to travel to the cell's protein factories, the ribosomes. Here, the mRNA's code is translated into a chain of amino acids, which folds into a functional protein. Each protein then performs a specific job, from digesting your food to fighting off infections. This elegant, step-by-step flow from DNA to RNA to protein is the central dogma of molecular biology, and it operates ceaselessly in every living cell. Yet your DNA is not a fixed text, it carries the echoes of your entire evolutionary history. Over 8% of your genome consists of ancient viral DNA, remnants of retroviruses that infected our primate ancestors millions of years ago. These sequences, once considered junk DNA, now play crucial roles in regulating gene expression and even in the formation of the placenta. Furthermore, your paternal lineage can be traced through the Y chromosome, while your maternal ancestry is written in mitochondrial DNA, passed down unchanged from mother to child. By comparing genetic markers across populations, scientists have reconstructed the Great Migration of Homo sapiens out of Africa around 70,000 years ago. Your cells are thus living archives, containing stories of pandemics, migrations, and adaptations that shaped humankind.